Okay. Hi, good evening, everyone. So today we will be continuing the session for Animal Physiology course. We will be picking on a new topic, which is about the visual system, which is about eyes. We will look into the anatomy of eyes, and we will discuss questions which are pertaining to the topics that you would have studied in the week 9 of the lecture series, right? Okay, so let's start. So this is the problem solving session 9 for animal physiology course, and in this we will be studying about the like we'll move ahead of neural system and we'll do special systems. So we'll begin with eye visual system, and if you remember, week 9 had these 5 lectures. You had anatomy of eye. You had just a second. Yeah, you had eye lens and cataract. Then we looked into the structure of retina. We then looked into image formation and processing in eyes. And the last one was mechanism of photoprocessing by rods. So these are the 5 lectures that you have looked into in the week 9. And these are the topics on which the questions of this problem solving session is based upon. Most of these topics are interrelated to each other, so you would often find that like the previous topic has parts of the upcoming topic, so they're all interweaved into each other. So I will also first begin by giving you a recap of all these 5 lectures, what was explained to you in these 5 lectures, and then we'll move on to the questions for each of these separate topics. Right? Okay, then let's begin. So as the topic is about eye system, this is about visual system. So if you look into the eye system here, what you see on your slide is basically the anatomy of the eye. So you see sclera, you see this pupil, you have iris. Now this is this portion of your eye. If you also look into further details of eye, you will see what we just discussed about sclera, iris, and pupil. They are here. Along with this, you have cornea, lens, ciliary body and muscle, conjunctiva, retina, which is very important, vitreous body, which is here, optic nerve, which is also very important, macula, which is here, and the blood vessels in the eyes. Now, if I have to talk about each of them in details, or if I talk about their function, so if you look into this, the first thing that you see about the eye, if you start like, so basically, if you have to define eye is a specialized sensory organ of the visual system, and they are responsible for capturing light. First of all, so you have the light which is incoming, you capture that, the eyes capture that, and then they convert this into neural signals. So the light that is coming is converted into neural signals, and then it is sent to the brain where it is interpreted as a vision. Right? Now, if you begin from the exterior anatomy, your eyes are protected by eyelids and eyelashes, which you can see. So this is the eyelid, this is the eyelash. We know that they protect the eye from dust and injury and excessive light. The next one is sclera. So the sclera is the white outer coat of the eye, which is a very tough connective tissue, and it basically is involved in maintaining the shape of the eye. Right? The next one is cornea. The cornea is the transparent part of the sclera. So you can see the sclera is here. This is the white part. Now, if you see the cornea, it's right in front of the like the front portion of the sclera. So what you know about cornea is that they are basically transparent part of the sclera, and also with respect to the image formation of eye, they are the first refractive surface that bend the light. So you all have studied how images form because of light, right? We have studied that in physics. So the same concept applied here. You need refraction of light. You need the light to bend to converge at a point. So the first surface where the light gets bent is cornea. Okay, now moving on to the next parts, you have the iris and the pupil. Now, what do these do? Iris and pupils. The iris is basically the colored part of the eye. So we all know that eye have different colors. So that colored is imparted by the iris, and these contain muscles that regulate pupil size. So basically, eyes surround pupil, and pupil is where you will see the light it enters. So the iris adjusts the size of the pupil. Okay, and now pupil, if you see here in this figure, the pupil is here, or the pupil is this. This is the iris, this is the pupil. So the pupil is the part of the eye from where how much light enters the eye is controlled. Okay, so you have pupil, which allows how much light would be entered, and that how much is determined by this iris contraction. We will see in the later slides, right? Now, after that, we move on to lens and ciliary bodies. Now, lens is a very common term that we keep using every day. So lens are basically the transparent, flexible structure which is behind pupil. So right after the entering of light, light enters through pupil and encounters lens, right? So this lens basically after pupil, they're behind pupil, so they fine tune the light and they focus it onto retina, which we will see that that is where the image is formed. So they take the light and they focus on the retina. So lens is basically fine tuning the light that is coming in, right? And the ciliary body or the ciliary muscle is helping it to focus the light onto the retina. Okay. Right. And then ciliary body are basically, it's the one that produces something called aqueous humor. So you saw here vitreous body, right? You also have aqueous humor, which is in front. So it's basically for the moisture of eye. It keeps the eye and controls the lens curvature, right? So we have aqueous humor and vitreous humor, which are basically fluid in your eye. So the aqueous is between cornea and lens, and the vitreous is between the or it is behind the lens. So how you can remember, you can see that this aqueous is A. So basically it is at the front and then comes vitreous V. So it is at the back. So between cornea and lens, you have aqueous humor. Between the or behind the lens, you have the vitreous humor. Right. And what do this fluid do? They basically, as I said, they maintain the moisture of the eye. Along with that, they also nourish the cornea and the lens, and they maintain the intraocular pressure. Okay, they do not let the eye go dry. Okay, if it is, then it's your eye shouldn't get dry, right? You need that water in your eye. So these humors are for those. Okay, now the last part which is left to discuss is the part which is here, present here, right? So the first part here is retina. So this retina is basically the lining at the back of the eye, and this part contains photoreceptors. As the name suggests, these are receptors for light. Now these are cells which are receiving the light. These cells are of two types, rods and cones. You would have definitely remembered from the lecture series. So rods and cones. Now what do these rods do? They are both basically they are receiving lights, right? So they are both of two different categories so that you can have the maximum light information available to you. So rods are sensitive to dim light, right? And they are mostly, of course, because it's dim light, it's mostly for night vision, for black and white coloration. Cones are for the colors and details, for day visions and high acuity. So how you can remember which cells is for what? Cones are C, so they are for colors, and the other ones are for black and white. So rods and cones. Okay, that's on the retina. So that's basically here where your image is formed, right? Now you have macula also. Now what is this macula? Macula is central retina. So this is part of the retina, but at the center position, and it is specialized for high resolution vision. Okay, and then you have a center portion of macula also, which is called fovea centralis, which is again in the center of macula, again for sharp vision, and which has more of cones. And then there is a very important part of a retina which is called blind spot. As we will see that we have optic nerves coming out of this eye, as you can see in the figure also. You have these optic nerves coming up, right? So the part of the eye or the region of the eye where you don't have the photoreceptors is where the optic nerve exits. So these optic nerves are exiting from here, right? And here you don't have the photoreceptors or rods and cones. Because you don't have rods and cones here, these place is called blind spot, right? So these terminologies are very like I told you, use in day-to-day life also. So I will next in the next slide we will see how can you remember it more easily if you compare it to an like what should I say, a device that you use in daily life. Okay, we'll see it later. Right. Other than that, what you should remember is in eye, you also have something called retinal pigment epithelium, which is so we'll see here when we go into retina in detail, but you will see there will be retinal epithelium, which is basically helping nourish, they're basically nourishing the rods and the cones. Okay. And the most important part is the optic nerve and visual pathway, where we will see that after the information is received by retina, it is taken by optic nerves to the brain. And how that entire thing takes place is when I will tell you how, like from retina when it moves further, right? So now I told you that how you can remember it with a common device that we use every day. So eye is doing the same function what camera does for you in day-to-day life. You have camera doing the exact same function that eyes does for you, right? We use camera for clicking photographs. Eye is basically doing the same. Now, how you can remember what eye does and how it is synonymous to camera? Let's put some of the parts of camera and parts of eye back in front of each other. Okay. So eye, like I discussed, have cornea and lens, which are basically functioning like lens in camera. So as you can see here, this is the lens here, right? Lens here. This lens is similar to lens in camera. Now, if you, some of you are more like you people use camera, then you would know then that a lens in your camera are basically for focusing the incoming light on a surface, and in your eyes cornea and lens does the same thing, right? Next is aperture of camera. If you know what's aperture, it's basically here you will, it basically close and shuts to like regulate the light. Which one does that in our eye? We have discussed it. It's all the iris and the pupil. So they control the amount of light that is entering your eye, right? So the iris can contract and relax, like I have told you, and to change the pupil size. Next is retina, which is in eye and is similar to the CMOS of camera. Now, what happens here is, as we know in retina, it is where your image is captured. So your image is focused on, the light's focused on to that part. So retina is where your image is formed, right? And then you have the photoreceptors in your eye and your pixels in your camera, which is basically interpreting the light that is coming. And then you have another terminology that you can remember is a shutter in your eyes, shutter in your cameras, which is basically the one that lens cap, which closes the lens. For that, you have the eyelid in our eyes, right? And the last thing, which is also very easy for you to understand, is that how do you transfer your images from your camera to your laptop? Via a cable, right? The cable takes in the information from the camera. So it basically is transferring data from the camera onto the storage or display, wherever you're putting. So it's relaying the information. The same way in your eyes, the optic nerve carries that information as signals to the brain, and the brain interprets this as an image. So you have all these, like if you place them simultaneously, you will see that one, it will help you remember the function. Second, it will help you also visualize that how camera and eyes work similarly, right? Okay. Now let's move into much more details of lens and retina and see into their features and why they are helpful and how they are important for our image formation. Okay. So let's begin with lens. So the first and the most important thing that you need to remember about lens is that they are transparent. Okay. They are located behind iris and pupil, and but in front of the vitreous body, which is around the vitreous humor. And they're transparent and they're flexible. This is also very important that you should remember. If you know, people use these lenses, people can change their lenses because, and they are able to do that because it's very flexible. So you can just basically just put it in your eyes. So you are basically, you can change your eye color because you're changing that color there, right? So what are you doing here? Is you are, so people use lens as a corrective measure, right? So your lenses are basically also doing the work of your spectacles, right? But what I want to tell you here is that lenses are flexible. You can remember the flexibility from this perspective that we can use lens, right? Now, let's look into the structure or the anatomy of lens. You have something called lens epithelium. You have something called lens capsule. Then you have a lot of lens fibers, and then you have the ciliary body. Okay. Now, what is this lens capsule doing? It is the outermost thin membrane. What is the epithelium doing? It is under the capsule, so it produces the new fibers. Now, what are the lens fibers? They are basically crystalline proteins. Okay, and they are transparent. So these crystalline proteins, as we will see in the question when we will discuss them. So these crystalline proteins are arranged in a way that they maintain the transparency of the lens fibers. Okay, and the most important thing that you need to remember about lens is they are avascular, which is basically means that they don't have blood vessels. So you don't have blood vessels. We will discuss why we don't have blood vessels and why is it needed like that. But for now, just remember that lens are avascular, lens are transparent. Okay, and if you talk about lens function, it is basically helping you bend the light onto the retina. It's focusing the light onto the retina, and it's doing so because of ciliary muscles. Okay, now how is it helping? How is the ciliary muscle action moving, focusing the light? So what is the change in focusing if the ciliary muscles change? So if your ciliary muscles, which are near the lens here. So if your ciliary muscles contract, your lens become thick. So just imagine here, if this contracts, so they are basically coming close to the lens becoming thick. So if your lens is becoming thick, why is it helping? It is helping you for near focus. So you can see nearby objects better. If your ciliary muscles relax, your lens will become flat and you can do far focus. So for seeing your far and for seeing your near, you generally use your ciliary muscle to regulate the amount of light that enters your eye. Okay, right. Okay, and why do you need transparent? If you can take a guess, it's very simple to understand. Why would you need a transparent lens? Because you want a clear path for the light to go. There should be nothing that like reflects the light off. If there's red color, if there's blue color, if there is anything else which is present in the process of entrance of light, then light will be reflected off. That you don't want. So for your light to go straight or enter without any problem, you want a transparency. That's why lenses are transparent in nature. Okay. All right. Now, if that's about lens, what's about retina? What do we know about retina? So let's talk about retina now. So retina, as you know, is the innermost layer of the eye, which is lining the backside. And functionally, this neural tissue converts the light signal into electrical signal. Okay. Now, the layer that I told you last time was retinal pigment epithelium, which is here, RPE, one of the most important layer. Then you have the photoreceptor layer, as the name suggests. These are basically the layer that consists of the photoreceptors, the cones and the rods. Okay, you can see the cones are colored in blue, green, and red, while the rods are for black and white as their functionality, right? Then you have these nuclear layers, which are outer nuclear and inner nuclear. So outer nuclear is basically this, which consists of the photoreceptor cells, and the inner nuclear are the one which consist of other cells, which are like bipolar cells, horizontal cells, and amacrine cells. Now, what are the function of these cells? We will see when we do the question and answers. Okay. And then you have the ganglion cell layer, which is very important, which is the ganglion cell bodies here. And the bipolar cell, I already mentioned. Now, these cells are important when we will look into the electrophysiology of the eye. Okay. And lastly, you have the nerve fiber layer, which is basically the axons of ganglion cell, which form the, so if you see here, these these ganglion cells are the one that finally form the optic nerve. Okay. Right. Now, if I talk about the function, we already know it has rods and cones for both light sensitive and dark sensitive vision. So your rods, which are for the dim light vision, they are called the scotopic vision. The cones, which are for color visions, are called are for the phototopic vision or the color visions, right? Now, these photoreceptors, they first, as you can see from the arrangement also, they will first pass on the information to bipolar cells, which will then pass the information onto the ganglion cell. So first information is received by photoreceptor. This passes to the bipolar, then it passes to the ganglion cells. From the ganglion cells, toward the end, it becomes the optic nerve. So from the optic nerve, then finally goes to the brain, right? Now, this is the layer, this is the pathway how your images are made by the brain, and it interprets these signals. What do these extra cells do? Amacrine cells and the horizontal cells. These are basically the giving the other information for your image generation. For example, contrast, motion detection, the edges, the fine resolution. So your signals not just give you like not just the light is being focused, the detailing of your image is being given by these other cells which are present in the eyes, right? So other than photoreceptor, you have these cells which basically enhance the detailing of the image that you are seeing, the clarity of the image that you are seeing. Okay, right. Now if we have done about lens and retina, then we can now understand what is cataract and what is cataract surgery. Now, as the image shows you, you have a blurry vision here. You can see a cloudy lens here. And here on the right, it basically is trying to show you to remove this problem. You basically do surgery where you are basically, so you can see. So this is the lens, right? Your lens is supposed to be clear. And if the lens is cloudy, become cloudy, you can understand. If it's cloudy, what I told you for lens, it has to be transparent so that light can pass easily, right? Now, if it's becoming cloudy, it can become problems, right? So that's why if it's if this is the condition, it's called cataract, and you remove it, that is called cataract surgery, right? In very simple terms. So why do you have this clouding of the eye lenses? This could be because of aging, which is the most common factor, because we told, right, that your lens is formed because of your crystalline proteins, and we will see that your crystalline proteins are arranged in a way that it does not lead to protein aggregation. But over the period of time, you have these protein aggregations in eyes. Simple to understand. Basically, the way you should understand these anomaly is that you don't want any hindrance in your pathway of light. So this protein aggregation is a hindrance. So it will not allow light to pass. So it's a problem. So in old age, you have this protein aggregation in lenses, which is leading to the cloudy appearance of the lens. Other than that, you have eye injury or trauma or any medication, or you have so cataract can also be congenital, which is basically it can be since the birth. Okay, so that could also be there. And you are basically also you can also have cataract if you have oxidative stress like UV, smoking, diabetes, UV exposure basically. So smoking, diabetes, UV exposure can also lead to cataract. So once you have these cataract, you have this lens formation, cloudy lens formation. There will be blurred vision. You will have sensitivity to light. Your colors will fade. You will start seeing very black and white kind of vision, double vision because you're not able to focus them. So you need this cataract surgery. Is basically removal of this clouded natural lens, and the doctors will then replace this with a clear artificial intraocular lens. So this is what is the image trying to show here, that there is a cloudy lens is removed, and then you have a clear artificial lens which is implanted into the empty capsule of the lens, and then this artificial lens is then acting and in place of your natural lens, and it is corrected, so your vision is also corrected after this. Okay, right. Now let's move on to how actually does image formation takes place in the eyes. We've already done this, so it's not very difficult to understand. Just we will look into uh, the formal way, or the way you can write it, or the words that you are using for explaining this. Okay, so the image formation of eyes can be basically divided into several steps. First is light entry, for which we know that the cornea, pupil, iris, and lens, they work together. So how does it move? It moves through cornea, aqueous humor, pupil, which is maintained by the iris. So pupil, then lens. From lens via the vitreous humor into the retina. That's the light entry, right? Okay. And at all these layers, your light is bending. So your light is refracting. Now, so you have cornea and your lens which bend these lights and convert on the retina. Now, remember, if you have specs on, you know that you are either a myopic or you are either hypermetropic. And those are, we call them as short-sightedness or long-sightedness. And that's because where your image is formed. Now, if your light gets focused here, which is your retina, remember the image which is formed on your retina is an inverted image. If you would have done, like the pictures where we have seen image formation, we know it's formed real inverted image. So similarly, eye also forms real inverted image onto the retina, right? And then this inverted image is corrected by brain, and you see the correct straight positioning of the object. But if your image formation is before, if your light is focused in front of retina, it is called short sightedness. If your light gets focused behind retina, it is called the long sightedness, right? Okay, like I said, it is real inverted in image, and also it is reduced in size. So the brain actually takes, so you can imagine what all the brain is doing. The brain is getting a small image signal to interpret, but the other cells are giving it all the other required information so that this small image is actually blown into the correct size, and it's being interpreted, and you can see them as vision. So all these cells in the eyes are important for working for the visual system that you see. It's not just the roads and the cones. Okay. Right. And then, so I already mentioned to you how this light is reaching here. And after this light is reaching here, from here, it goes to the brain. So from the photoreceptor cells, bipolar to ganglion to nerve, and nerve to visual cortex of the brain. So where does it go? It goes to the visual cortex. Now, as you would have remembered in the lecture, Professor talked to you in detail about the electrophysiology, which is basically the hyperpolarization and the depolarization of the or action potentials in the eyes, which is why you have the signal which is growing to the brain, right? So I will just quickly tell you, not going into too much time into this, because in some question or the other we will encounter this. So basically, what you need to remember is this ERG here is basically your electroretinogram, as like electrocardiogram is basically telling you how your light and how under light and dark conditions the action potential varies in the eye. Now, the image is talking about several steps. We are not interested in that. Let me just tell you the basics of what happens in the eye. If you remember, the professor very nicely told you that unlike other conditions here at the resting stage, ideally in resting stage, you don't have depolarization. You don't have depolarization, right? And resting state means everything is normal. But here in eye, in resting state, you have the sodium influx. Right? Remember when did we say sodium influx? You have depolarization, and we always say at resting phase you have the like your cells are not depolarized. But here in eyes, in resting phase, your cells are depolarized. What and how? So basically, the resting state for eyes is dark condition. And in dark condition, your rods and the cones, which are basically the photoreceptor, they are both depolarized. Depolarized how? Depolarized because there is a sodium influx. So your sodium channels, which are in the outer layer, they're open, and the sodium influx is happening, right? And this is happening because of two reasons. First is your sodium channels are opening, and they're opening because of a protein called CGMP. So you have a lot of CGMPs which are keeping these channels open, and your sodium is moving in, and the sodium is moving in also because of the release of glutamate. So your bipolar cells, which you remember are just above the pros and cons, these bipolar cells are releasing the neurotransmitter saying glutamate. So the glutamate has been released, and your sodium is getting influxed. So this is the state where light hasn't entered. In dark condition, your cells are depolarized already. Okay. Now this is resting. What happens? So the thing that alters this is light activation, which is basically when light enters cells. So when your light enters cells and hits the retina, protein in the retina is called opsin. So when it hits the opsin, the protein itself undergoes a change in configuration. There is cis configuration moves to a trans configuration. So that change in the configuration from a cis to trans activates that protein. A protein opsin gets activated. This induces or activates transducin. This transducin activates phosphodiesterase. Let me say this once and then I'll come back to it again. This phosphodiesterase hydrolyzes the cGMP. Now, what was cGMP doing? cGMP was keeping your sodium channels open. So, as soon as upon light, your cGMP are basically hydrolyzed or basically removed away because of phosphodiesterase, your sodium channel is now closed. Now, as soon as your sodium channel closed, your glutamate release also stops, and that's because now your cells have undergone into hyperpolarization, because remember, sodium influx is making it depolarized. Sodium influx stopped, it has become hyperpolarized. Okay, let me just revise this 1 and 2 again. So at resting phase, which is dark condition, sodium channels are open, sodiums are moving inside, sodium ions moving inside. Cells, which are bipolar cells, are releasing glutamate, so they are under depolarized condition. Now, when lights hit retina, the protein opsin undergoes a configuration cystotrans. As soon as that happens, what it does, it initiates a cascade of reactions. The first reaction it basically does is first itself gets activated. Then it activates transducin. That activates phosphodiesterase. You need to remember the last protein, phosphodiesterase. The diesterase means it's an enzyme which is cleaving off the ester bond in cGMP. cGMP is hydrolyzed. cGMP was keeping your sodium channels open. So cGMP destroyed, sodium channel closed, cells undergo hyperpolarization. Okay, now this is your depolarization and hyperpolarization, right? Now, when your glutamate cells are, so the bipolar cells, as we know, if they are depolarized, so when they are on bipolar cells, uh, they are basically depolarized when the glutamate condition decrease, and they are off when it is hyperpolarized and the glutamate condition, like if they're hyperpolarized, they're off. If they're depolarized, they're on because they're releasing the glutamate. Okay. So as soon as your light enters and the cascade leads to the reduction in glutamate, this signal is given to ganglion cells, and ganglion cells, upon receiving this signal, when there is an from the bipolar cell, there is a change in the glutamate secretion, that cells generate the action potential, and these action potentials in the ganglion cells uh, reach the axons. The axons, as we know, make an optic nerve, and via optic nerve, it reaches the brain. That's it. That's what you need to remember. Okay. So dark cycle, light cycle, bipolar cells to ganglion cells, and via that to optic nerve. This is the electrophysiology of the eye. Okay. Right. Now, this figure here is basically telling you what I just told you. You have light. So you have the rhodopsin and the retinal, which is getting activated. It is activating transducin, which is activating your diesterase, which is breaking off your cGMP. Phosphodiesterase is breaking off your cGMP. Your sodium channels close and your cells undergo hyperpolarization, right? So photoreception is basically the process that describes the electrophysiology of the eye. Okay. So this basically covers up everything that you have done in the week 9. And now subsequently we have questions, and I will read out the question, wait for a few seconds and then give you the answers. If you are attempting back to your system, you can pause the video after my questions, attempt the question and then replay, and you will have the answers and the justification for the answers as well. Okay, that way you will be also solving along with me, and that will help you know. And I hope the questions help you understand these topics better. These are simple topics to understand, and like I said, they're all interconnected to each other. So once you have clarity about one topic, it becomes it's like a story. It's like a flow chart. So you read the entire topic in a go. Okay, maybe cataract is the only topic which is anomaly to the normal functioning. So I've kept cataract at the end. I've just first covered the entire flow chart process one by one, and then we move on to the cataract. Okay, right. So which of this is not part of an eye? Iris, cornea, hair cells, and ciliary muscles. And the liquid filled between cornea and lens is called what? And second part is, whereas from lens to retina is called what? So the first question is basically simple anatomy question, which basically asks you which is not part of the eye. The second question asks you which of, so between cornea and lens, And between lens and retina, which part is present? So I will wait for a few seconds for you to read the question. Right. So let me just give you the answer for this. So the first one is hair cell, which is pretty easy to understand because we have already discussed about iris, cornea, and ciliary muscles. The next one, I told you a cue for you to remember this. So the liquid filled between cornea and lens to retina, you can remember it in the way of the anatomy. Whichever part comes first, that has aqueous humor. A comes first, so that part, so the anatomically which parts come first, it has the aqueous humor. The next part has vitreous humor. So which part comes first? Cornea and lens. Cornea and then lens. So you have aqueous humor. Lens to retina, which is vitreous humor. Right? So most of the time, these topics, as I keep saying, these look very humongous and big. But if you put some small keys to it or logic to it and try to remember it, it makes your task very easy. Okay, right. Now, which part of the eye functions similarly to the lens of the camera? Now, if you remember, this is what I wanted to like tell you when I was comparing lens and camera, eyes and camera, right? So basically this question asks the same thing. So maybe when you are done answering, we can just go over once again, what's the similarity between lens and eye lens or eye and camera. I'll wait for a few seconds. Right. So which part of the eye similarly works similarly to that lens of a camera? The lens, right? This is easy to understand. But let me just tell you, so the four functions that we have here: cornea, retina, lens, and optic nerve. So the lens of eye, why is the lens of the eye similar to lens of the camera? Because the lens of the eye focuses the light rays onto the retina, just like camera lens focus light onto the sensor of the camera. Right. Now, what does cornea do? Cornea, as we know, that it is help in refracting the light, bending of the light. So in the lens of the camera fine tunes the focus, right? So it doesn't help in bending. So what we mean here is by lens does not bend the light. Lens, so cornea bends the light, lens help in focusing. Okay. Retina is the final sensor, which is not what lens does. Lens also give it to the sensor of the camera. And optic nerve, as we had discussed, it basically takes the information away from the device. So for camera, optic nerve will be equal to the USB cable that you use for transferring data. Okay. All right. Next one. Which of these enzymes, if linked with, is linked with formation of ice? Not if, is linked with formation of ice. Eye lens, sorry. So you can, so basically lens is, like I told you, it's an aggregation of crystalline proteins. So what you need to remember here is what is helping in the like the arrangement of the crystalline proteins in your eye lens. So after every question, I will just wait for 10 seconds so that in the video also you can pause and come back. Right? So the answer here will be 15 lipooxygenase. Okay? Right. Now, I told you eyes, the lens of the eyes are transparent. Okay. Why is it transparent? Why is the lens of an eye transparent? Is it because of the presence of pigments? Is it because they have highly ordered arrangement of crystalline proteins? They have high blood flow, or they have thin epithelial layers? Okay. So the answer is highly ordered arrangement of crystalline protein, as I have been telling you so far. But let's now look into the reason why this is the correct option and the others are incorrect option. So these crystalline proteins, they are highly ordered. Highly ordered means they are stacked with each other, right? So they are not scattered. They are all compactly packed with each other. Okay. And that makes it like a regular array. Now, what happens? Why should it matter? Because I told you the whole idea about having a clear path is basically to allow the light to pass through without any hindrance, like unhindered, right? Now, if your crystalline proteins or any protein in your lens for that matter is everywhere, so you have blobs of protein here, then here, then here, then here, then here. So it's making a lot of gaps, right? And if these proteins are the ones that are to pass, that are making up the lens, you can see how gappy the lens will be. So you want everything packed together so that the lens forms just like this and it just passes through it, right? So you want this compact arrangement of the proteins, which are the crystalline proteins, right? So that they don't scatter light because they are here. If they are, let's say, these are the two crystalline proteins and they are here, one is getting light here. It is supposed to refract light, so it will bend light here. This will bend light here. So it will all go haywire. If they're all stacked together, they will all minimize the scattering of the light. They will all focus the light together onto the retina. That's the whole idea, right? Now, this is the correct answer, but then what is the problem with the other answers? So the first one says that presence of pigments. Now, I told you the whole idea of not having a pigment is they would either scatter the light or absorb the light. So you don't want that. You want the light to move through, right? These don't allow anything which has pigments has color, and that is basically not transparent anymore, right? High blood flow. If you remember that the lens are avascular, they don't even have blood vessels, so that you don't even want blood vessels to obstruct the light. So you don't, the blood vessels also are categorically removed, or you will see that it is moved away from the crystalline protein cells, eye cells, right? And then still epithelial layer. This is basically layer which is responsible. So the eye lens is thin, but that doesn't ensure the transparency of the eye, right? Or the lens. So transparency is maintained because of crystalline proteins, right? Okay. Moving on. Now, next question is during lens formation, like I was just saying, program cell death is important. So I haven't discussed this because I told you it will come in question. So first, let me wait for you to read the questions. The question says, during lens formation, program cell death, which is basically apoptosis, takes place. This is important because it reduces the number of lens fibers, removes the cell nuclei to create an optically clear lens, prevents cataracts, or thickens the lens. Right. So I hope you understood the question. So the answer, first of all, is it removes the cell nuclei to to create an optically clear lens, which is basically, so when your eye lens development is taking place, the epithelial lens which elongate to become the lens fibers, right? Now in these fibers there is apoptosis of the nuclei and other organelles, right? Now what do you understand by it? That you have these cells who have the nucleus, who have the other organelles right there. These organelles and cells are removed. Now, in either condition, this would lead to the killing of cell. The cell will also die. The apoptosis means cell death. But here in this case, they're categorically removing the nucleus, categorically removing the organelles so that the lens has no other proteins except for crystalline proteins. The cell remains viable. Okay, and it's transparent because everything else which could have hindered the light movement is now removed, right? So basically, just retention of crystalline protein is what making the eye lens transparent. So your cell death is to make it clear, not to reduce the lens fibers or prevent cataract or thicken the lens. Those are not the primary, or these are not related to apoptosis. Okay, next one. Similar question in other terms so that you get well versed with these different topics. So self-assembly of a alpha crystalline protein in the lens. Self-assembly is basically you don't need any other things to come and bind to it, right? So you have this alpha protein, alpha crystalline, which is assembling on its own in the lens. So I told you lens is clear because of these how do you say ordered arrangement of crystalline protein. But why is this protein is written? Why is it important? That's the question here. So is it important because it provides energy for lens contraction? Is it important because it maintains lens transparency by preventing protein aggregation? It is important because it transports ion across the lens membrane, or it is important because it serves as a neurotransmitter. Right. I hope you understood the question. The answer to this is maintains lens transparency by protein aggregation. So this is where you will realize what is the importance of the protein alpha crystalline. So this alpha crystalline protein, uh, they self-assembler, they order themselves, they arrange themselves, they aggregate. So if they aggregate, it leads to the formation of cataract formation. We'll see why. So alpha crystalline basically is basically a molecular chaperone. Okay. Now, what is so the crystalline protein is a heat shock protein, and it is basically a molecular chaperone. Chaperone is basically something that aids other protein. Okay. So this protein, crystalline protein, alpha crystalline, is a molecular chaperone. It binds to unfolded proteins or the partially folded proteins, and they don't doesn't let them aggregate, right? So they are nicely arranged. They also help these other proteins which could possibly, now imagine this is a lens, everything else is removed, and you have these small proteins which are starting to aggregate. So these crystalline proteins will bring these proteins also, which are half aggregated or partially folded, and bring them and put them together so that nothing disrupts the pathway of light. And that's the whole idea about transparency, right? So that's why these crystalline proteins are important because they are molecular chaperones. Okay, right. Next one. Okay, now this is what was the whole idea here, that the unique folding pattern of alpha crystalline, which allows it to do what? Does it allow it to bind tightly to DNA or function as a structural molecular chaperone or generate action potential and facilitate lens hydration? Which of these do you think is the answer based on the discussion that we had just have? We just discussed, right, that the answer to this will be functioning as a structural molecular chaperone. Okay, let's move ahead. Now, the cornea, which you know is present as a first layer, it differs from lens in what terms? So, read the options. The cornea is flexible and the lens is rigid. The cornea provides most of the eye's refractive power. The cornea is vascularized while the lens is not. And the cornea focuses light dynamically. So which of this is, so basically whenever it says cornea does this, it means the lens does not do this. So what is true? Which is the difference between lens and cornea? Read and answer. Uh, this is not difficult, but just read. You should be able to do it. Okay, let me give you the answer here. So the cornea provides most of the eye's refractive power. Let's do the other options first, and then I will come to this. So first option says cornea is flexible, lens is rigid. Do you think this is correct? This is not what we discussed. We discussed lens is flexible and cornea is rigid, right? Next option is the cornea is vascularized and the lens is not. Well, lens is not is correct, but you should also know that cornea also is avascular. That also does not have blood vessels. So both of them are without blood vessels. So they are both similar in this pattern. So this doesn't make them different. Next one says the cornea focuses light dynamically. Cornea doesn't focus light. Cornea refracts or bends the light. Focusing of the light is done by your lens, right? So it is an incorrect statement. So the first statement, or the second statement, which says that cornea provides most of the eye's refractive power is true. So you would say that lens also refracts, yes, but the word here is most of the refractive power, which is correct because your cornea has does the 70% of the refraction and your lens does the remaining 30%. So the statement is correct in the terms that it says mostly it is being done by cornea. That's why it is the correct statement, and that's the difference between cornea and lens, right? Okay, next one. Now, easy question. Which part of the eye contains photoreceptor cells, which are basically rods and cones? But I will wait because I have been doing it so that we come to the same term. And we already know the answer to this is retina, right? We have already discussed this. Okay, now, which retinal layer contains the nuclei or the photoreceptor cell? Basically, where do you have the photoreceptor cells? Inner nuclear layer, outer nuclear layer, ganglion cell layer, and retinal pigment epithelium. Right. So if you remember, the answer to this is outer nuclear layer. Okay. So nuclear layer, inner nuclear layer, and outer nuclear layer. So then what does the inner nuclear layer have? They have all the other cells, if you remember, bipolar, horizontal, and amacrine cells. The ganglion cells have the ganglion cells, like the ganglion cell layer have the ganglion cells. And the retinal pigment epithelium, they basically have a pigmented cells, as the name suggests, they have pigmented cells and they support the photo. They are mostly for nourishment and your, like maintenance of your photoreceptor cells. Okay, these are present in the outer layer, nuclear layer. Then you have the inner nuclear layer, which is basically having the other X cells that we have. Okay, now retinal pigment layer only. So we have now there are two features here. One is saying that it provides nutrition to rods and cones. Second says that it prevents rods and cones from photo damage. So which of the two statements are correct? Right? And I just discussed that the retinal epithelial layer does both of this activity. It also provides nutrition and it saves these cells from photo damage, which is basically damage from excessive light. So it protects these cells from excessive light. Okay. Coming back to this layer again, the RPE. Right, I just said. What does it primarily do? Does it primarily generate action potentials? Does it absorb stray light and nourish photoreceptors, store neurotransmitter, and synthesize alpha crystalline? Why is this seem repetitive? Because I've just explained to you. But this gives you the exact word why it prevents photo damage. And as you would have already guessed, that these are responsible for absorbing the stray light. I said, right, excessive light could damage your photoreceptors. So the excessive light is absorbed, or stray lights which are coming from here and there, they are absorbing them. So that's saving the or the photo damage protection. And the other was nourishment, which is what is written here. Okay. Okay, moving on to the next one. There are two questions. So I'll wait for a longer time here. The first one says, what is the primary role of retina here? Is it to focus light onto cornea? Read them properly. These options are very similar, but read them properly, then it will help you understand. It helps convert light into neural signals. It adjusts the size of the pupil, or it protects the eye from dust. The second question says the optic nerve is responsible for focusing light onto the retina, transmitting visual signals to the brain, controlling eye movement, and maintaining intraocular pressure. I'll wait for 30 seconds, a bit longer here. Right. Staying to a schedule. So what is the primary role of retina in the eye? As we have so far seen, it converts light into neural signals, right? And the optic nerve is taking those signals to the brain, right? Now, just for your confirmation, what are the other options saying? This one says focus light onto cornea. This is done by lens. Uh, yeah, this is done by lens. I mean, it should be retina, but it's okay. It's not the retina that focuses the light, right? Adjusting the size of the pupil is by iris, and protecting the eye from dust is eyelashes and eyelids. Okay. Now the second one, focusing light onto retina, we know is done by lens and cornea, right? Controlling eye movement, it's actually by nerves. So you have some cranial nerves which does this. And maintaining an intraocular pressure is something that we had done in humors, right? Aqueous humor. So this is there. Okay. Next, again, we'll wait for a few longer time here. The primary function of rods and the cones here. So the primary function of rods. So you need to remember which type of vision is associated with these cells. So the primary function of rod in the retina is to detect color, provide high visual acuity in bright light, detect light intensity and function in low light, which is called scotopic conditions, or transmit signals to the optic nerve only. And cones in the retina are responsible for night vision, color vision, and high acuity daily visions, producing melanin and maintaining the blood retinal barrier. Right? So I will wait for 30 seconds and then I can discuss. Right. So the two options, correct options are C and B. So as we know, as I've already discussed with you, rods are basically highly sensitive to light, right? And so they allow for dim vision or nighttime visions, and they are not for detecting color. So color, as we know, is therefore, if rods are for your dim light vision, colors are for your cones, and they are transmitting to so like they do transmit the optic nerve signals to the optic nerve, but the question here was primary function. That's why it's about detecting light intensity and then passing it on to the uh, optic nerve. Now, the cones, on the other hand, will be the cells which are responsible for bright light conditions. They will perceive the color, the sharpness, the detailed visions, and these melanin production and the blood retinal barrier. These are not rows of cones. They are responsible for vision. Okay. Okay. Next question. Which of the following cells transmit visual signals from the photoreceptor cells to the brain? Which is again simple question. We've done this already. Simple, right? It's the ganglion and the bipolar cell. So, like I said, the rods and the cones, which is the photoreceptor cells, they perceive the light information, and from them, the which give it to optic nerve, right? The RPE cells are for nourishment, And the amacrine cells are for your processing, like they will give you the contrast, they will give you the sharpness, resolution, but they are not for your visual signal transmission. Okay, now this also I had discussed, but it's like for your revision. So the synaptic terminals of photoreceptor release, in other words, which is a neurotransmitter in visual system. And we know that, right? Glutamate. We've discussed this. Okay, moving ahead. Now, the blind spot, this also I had discussed with you. The blind spot in the retina occurs because, is it because there are too many rods in that area? It is where the optic nerve exists and that's why it lacks photoreceptors. It is a region of excessive RPE, the epithelium cells, or is it the region where cones are absent but rods are abundant? Remember what I told you about blind spot and then try to answer this question. You remember, right? The entire discussion about the exit of optic nerve and that region having no photoreceptor. So it's basically your optic nerve is exiting. So it's a gap there. And because it's a gap there, there is no photoreceptor there. That's why you are blind, because you're not able to detect any visual signal there, right? So it's blind spot. You can remember it by saying that, like blinds cannot perceive color or perceive visual signal. This spot cannot perceive visual signal, and that's because it doesn't have the photoreceptors, right? And why? Why doesn't it have photoreceptor? Because it gives a gap for the optic nerve to exit. All right. This also I told you. The primary area in the brain which is responsible for visual information processing. Which of this cortex or which of this part? This is very simple, right? This was the visual cortex, or also located in the occipital lobe. Now, we know what is visual cortex doing. So how do you define the function of visual cortex? Does it control eye movement? Does it interpret visual signals into images, contrast, and motion? Generate light sensitive proteins? Or does it maintain the retinal structure? This one is also fairly simple. It's interpreting visual signals into images, comes from the light. Contrast and motions come from the accessory cells, right? Amacrine cells, bipolar cells, horizontal cells, right? So then let's look into the function of amacrine cells. So the amacrine cells in retina, what is the primary function? Are they responsible for transmitting signals directly to brain? Are they responsible for modulating and integrating signals between bipolar and ganglion cells? Are they responsible for absorbing stray light? Or are they responsible for producing neurotransmitter for photoreceptors? Read them properly and then answer. So do you remember? We discussed this about amacrine cells, that they are responsible for integrating these signals, right? Now, how can you remember these cells? You can remember that these cells are interneurons. So they are interneurons. So they are basically present between two cells. So they're present between bipolar and ganglion cells. So they basically integrate the signal from the bipolar cells, which is already receiving signals from the rods and cones. And these cells, along with the rods and cones, also give information like contrast, motion detection, temporal sensitivity, all these accessory information to the ganglion cell that further passes it on to the optic nerve, right? So it's an interneuron cells. I mean, it is an interneurons. So it's between the two cells and integrates these cells information from the bipolar cells onto the ganglion cells. So it's an interneuron, and we know interneuron does not transmit cells directly signals to the brain. Absorbing stray light, we have already discussed, this is done by the epithelial layer, and producing neurotransmitter is, again, as we discussed, it is not part of these cells, right? Okay. So next question. Now, contrast in visual images that you see, which is, so it is enhanced by direct photoreceptor activity, horizontal and amacrine cells interactions, refractions in the lens, or increased brightness only. Easy question, right? We have already discussed it like multiple times now. Horizontal and amacrine cells interaction. Okay, next part. Retinal ganglion cells, which is the cells which lead to optic nerve. So these cells are defined as photoreceptor that detect light sensitivity. So you should see their position and try to answer every time. Okay, they generate action potential that travel via optic nerve to the brain, absorb excess neurotransmitters, and serve only a structural role in the retina. Easy question actually, even if you see it that way. We know this already. These are the cells which are responsible for generation of action potentials because of all the light activation on retina. When there is a light falling on the retina, you have all these cascade actions going on, and that ultimately leads to your cell hyperpolarization, which leads to the generation of action potential, which is then further passed on to the brain. Okay, so now the subsequent question required little bit of thinking. I mean, it's not tough, but just wait and try to understand an answer. So I will wait for a few seconds and then we'll discuss these answers. So in bright light, rod are highly active, saturated and become inactive, inhibit cones from working, and depolarizing ganglion cells directly. So they are depolarizing the ganglion cells. What will happen if you depolarize direct cells, right? So think of this and then answer. Okay. Yeah, options seem clear, like nearby to each other, but the correct answer here would be saturated and become inactive, right? So as we know, rods are important for your dark vision or your dim light vision for your black and white color, right? So if you have extremely bright light, so the protein, the opsin protein in rods are called rhodopsin. So if there is extreme light, these rhodopsin proteins are fully bleached. Bleached means they become non-functional. So basically they become inactive, and because there is no functioning of rod, the cones take over, right? So they themselves become inactive, and because they are rendered inactive, your cone cells take over. So they are not active, and they don't inhibit cones from working because they are inactive. They have no roles to play. So they become saturated completely, and they become a non-functional act totally. So they don't depolarize ganglion cells, or they don't inhibit cones from working. They simply become inactive, right? Okay, so do you have any idea about the absorbance of rods? Okay, so basically it means with what wavelength do they absorb light? Any idea? So the correct answer would be 500 nanometer. Okay, so we did bright conditions. Let's do light cycle. What is cycle? I will not give you, so that I want you to read and try to answer. So during the light cycle, photoreceptors, they depolarize and release more neurotransmitter. hyperpolarize and release less neurotransmitter, fire action potentials independently, shut down completely without any activity. Read each of these and then try to identify which step is called the light cycle and then remember what happens there. You remember, right? The answer here is hyperpolarization, releasing of less neurotransmitter, because we remembered, right? The resting stage is when they are depolarized. When light comes, the entire cascade, if you remember, light comes, your opsin gets activated, it moves from a cis to trans configuration, gets activated, activates the transducin. Transducin activates the enzyme for diesterate. Phosphodiesterase basically cleaves or basically inactivates the CGMP. CGMP was the protein which is responsible for opening of the sodium channel. So when your CGMP is inactivated, sodium channel is closed and your sodium influx is stopped, which basically lets the cell into hyperpolarized stage. And when it was depolarized, the cells were also releasing neurotransmitter, which was glutamate. So as your cells undergo hyperpolarization and your gates close, your glutamate release is also decreased, right? So all this entire song and dance is happening because light is falling onto retina, and this falling of light is called light cycle, and this entire process, as we know, is called phototransduction, right? Okay. So if this is light cycle, what is dark cycle? Read and then read the options and then try to answer. So I'm sure you would have got the answer already. So the answer is depolarized and continuously releasing neurotransmitter. So just to highlight, the neurotransmitter are released by the rods and the cones to the bipolar cells, the cells which are right above them in the arrangement or in the structural positioning. So the rods and the cones, they release the glutamate onto the bipolar cell. And when they stop or when this release is impaired, that's when the bipolar cells get relay the signal to the ganglion cells, right? We saw the cascade there. So the information starts from the rods and cones. Okay, right. Okay, now in low light conditions, which is a darker environment, the image formed by the eye would be high in contrast, poor contrast, relying mostly on the rod activity, is unaffected because cones can compensate or become sharper due to increased pupil size. Which one of this is correct answer? Read and then try to answer. Becomes very easy if you are a photography person or if you have taken any image from camera. It's the same functioning here. We are talking about low light conditions. So we know that in low light conditions, the photographs that we take are of poor contrast. And that's exactly what happens in eye also, because in eye, in low light condition, only the rods are acting upon, and the rods have low spatial resolution that they cannot discern the positional information or the spatial information very distinctly. That's why you don't have a very clear contrast. You have a poor contrast. That's what in your when you go out in dark, your vision is very blurry and you don't have clear information about what lies ahead of you. Okay. Okay. Now, foreground objects, which are basically objects which are in front of you, what happens to them in visual perception? Visual perception is basically your interpretation of the objects by the brain. So are they processed faster than the background details, or are they ignored in the favor of background details? Are they ignored entirely by the retina or always out of focus? Think and answer. This is very interesting question. You see images daily, right? So what do you think the brain would be preferring to do? No guess there. I think it would have been fairly easy for you to guess that the answer would be it is processed faster than background, right? Now, why is that? Because the brain, like us, also perceives these foreground images or the foreground objects as like more relevant, behaviorally more relevant. So let's say you are walking or obstacles which is something in front of you. So what how would you see a background image? Imagine you're standing out somewhere and you are looking into a vast expanse. There is a tree and there are a line of trees there. So the trees which are in back, they are processed. You do see them, but what you see clearly are the images in front, the trees, the bark that are in front of you. That's because brain considers that these are foreground images and they are of more relevance to the like for us. So it will interpret this better in contrast to the background. So the background images are processed at a lower space or lower speed. The foreground images are processed faster. The contrast is also in the favor of the foreground object, the salience, the way the image is perceived is in the favor of the foreground object because it perceives it's more relevant to the eye than to the the foreground object is more relevant to the eye than the background object. So they are processed faster than the background object. Background objects or background information are processed, but at a lower priority. The priority is more to the foreground objects. Okay. Okay. Next one. Okay. So oxidative stress in the retina can lead to, so if there is an oxidative stress, which is basically excessive function, excessive light. So what happens? Does it enhance visual acuity, photoreceptor degeneration and vision loss? It increases the synaptic activity or it hyperpolarizes the RPE cells. Similarly, if you have fatigue damage in photoreceptor, which is fatigue is basically overuse. So if you have fatigue damage in photoreceptors, why is that? Why is the fatigue damage coming? Is it because of mechanical trauma? Is it because of oxidative stress and excessive light exposure? Is it because of high intracellular potassium levels, or is it because of excessive neurotransmitter release? Easy question, shouldn't be very difficult to answer. It just follows logic. I mean, I just told you fatigue damage is basically overuse. So why would you use the photoreceptor cells? If you use that logic, the second question becomes very easy. And the first answer, I think everyone knows already, right? So the answers are photoreceptor degeneration and vision loss and oxidative stress and excessive light exposure. Let me explain the second one first. Now, I told you fatigue, we all know actually fatigue is basically when you have overdone something. So what are rods and cones supposed to do? They're supposed to interpret light. So them having extra exposure, overexposure to light, and they're working over, they're generating more of these chemical species which have the oxidative. We have discussed oxidative stress in the previous sessions. So they have more oxidative stress, and over the time of uh, over the time, the extra use or overuse of any of on any object for that matter leads to its wear and tear. Similarly, if you have overuse of the rods and cones, they start degenerating, they start getting problem. So first they have damage, fatigue damage, and then they over the accumulation of the damage leads to degeneration, and because the photoreceptor is degenerating, they leads to vision loss, right? Okay. So the what is the overarching, how will you summarize this part? photoreceptors are responsible for really, see, why are these questions important? Because they use terminologies which are very similar to each other, but then the answers are, one of the answers are correct, right? So you need to read and really break the options apart and then tell which one is correct answer. So photoreceptors are responsible for relaying electric signals directly to the optic nerve, or they are converting light into electric signals through phototransduction. They are synthesizing neurotransmitters in the brain, or they are filtering out UV light. We've already discussed this multiple times, so it is not a difficult question. It just requires you to read them a bit carefully. Right? So photoreceptors are responsible for converting your light into electrical signals through phototransduction. Uh, photoreceptors are not the ones who are generating the electrical signals to the optical nerve, right? They are the ones which are generating the electrical signals and passing it on to the bipolar cells. This is the difference here. And they synthesize neurotransmitters? No, they release neurotransmitters, right? So that's not the one that are response. They're not making up the neurotransmitters there and not the filtering out of UV light. They're the primary role, as we have discussed. They take the light and they convert it into electrical signals. Moving ahead. Now, the electrical current generated in the photoreceptors in the dark is called what? What is the name of that current that is generated in the dark? Action potential, receptor potential, synaptic potential, or hyperpolarization current. So why is this important? Because we know that this visual system, the electrophysiology of the eye and the visual system is different from all the system that we have been so far reading. We know that action potential is the current which is generated when the cell depolarizes. But the answer to this question is receptor potential. So in the dark, even though the cells are depolarized, this is not a classic action potential, right? This is rather a receptor potential or a graded potential, right? So as we know, in the dark, the photoreceptors are depolarized and there is continuous release of neurotransmitter. So this change in the membrane potential here is called receptor potential. And this is when the cell is depolarized. When the cell gets hyperpolarized, this reduces. And that time your ganglion cells generate action potential, which goes to the brain. So action potential comes later. The first current, which is the dark current here, is called the receptor potential, right? Okay. Okay, now adding on to this, if you remember, what is the resting membrane potential of photoreceptors? Try to remember from the class, from all the like the images that professor had drawn onto the slides. If you remember, it was minus 40 millivolt. Okay, now we come to phototransduction. Now, if you remember, in the darkness, the configuration of retinal in rhodopsin, basically in rhodopsin, also the center which receives the light is retinal. It undergoes a change in the configuration, right? But what is the configuration before it receives the light? You remember? It's cis, right? So what change happens to cis, or what change happens when there is photon absorption, which is also the word for light reception or light absorption? How does the retinal, which is the activation center in the rhodopsin, change its configuration? Easy to understand, but why do you know what happens there? So we know that the change is cis to trans. So what happens is in this um, retinal, the rhodopsin uh, consists of, like I said, rhodopsin. The name suggests rhodopsin because it's an opsin present in rods, right? So the opsin has a retinal. That retinal is in cis position. So when you have a photon, which is absorbed, which is basically a light, when it strikes your retinal, it basically isomerizes the cis retinal to a trans retinal, right? And this happens in a very distinct position. So every position on the retinal protein is in trans position, except for one position, which is the 11th position. So on the encountering line, or when the light is fell, then it falls upon the retinal, the 11th position also undergoes isomerization and forms a trans position. So everything is trans now, and that's when it the conformational change happens in this opsin, and it activates this opsin, and then it leads to the entire cascade, right? So what is this cis and trans? It's basically that one position that changes its configuration and becomes trans. Right. Next one. The phototransduction cascade in rods involves the activation of which G protein? So G protein comes from the name of GPCRs. So G protein is involved in phototransduction. So basically, how do you remember this is just remembering the name and associating that with the correct classification. So I will wait till you read and try to guess. So if you remember the cascade, so after the activation of rods, you have the activation of transducin, right? So the rhodopsin activates your transducin. This transducin is a G protein. And then this transducin further activates the enzyme phosphodiesterase, which hydrolyzes the cGMP. Okay, so this is easy. So coming back to here again, phosphodiesterase activated in the phototransduction cascade serves to do what? We have just discussed it. I will just wait for you to read it and then attempt the answer. So we know the answer is hydrolyzed. They don't synthesize cyclic cGMP. They don't open sodium channel. That's the opposite of what they do actually. And they activate rhodopsin? No. Rhodopsin activates phosphodiesterase, is the opposite, right? Okay. Now in the dark, read this and analyze this and then try to answer. In the dark, cGMP concentration in the photoreceptor will be what? Read and then try to answer. Fairly simple. Just you need to just focus on what exactly happening in that phase. And we have discussed that it would be high because it is needed for keeping the sodium channels open, right? And so what is basically happening is when you have the sodium channels open, you have the sodium ions moving inside. That influx of sodium ions is which is called the dark current. So, and that's happening because there is continuous release of neurotransmitter glutamate. So it's actually maintaining your cyclic cGMP concentration at high end, right? Now, next part. Again, read and try to answer. The hyperpolarization of rod cell membrane upon light exposure occurs because of what? It's convoluted, but it's basically asking the same kind of question. So read and answer. All these questions will basically help you. So in different ways, we are asking the same kind of procedure. So it will basically help you in understanding the concept to a very, very detailed manner, right? Oh, so coming back to the question, hyperpolarization is the next step after depolarization. So in this step, we know the sodium channel closes and your sodium influx is reduced, right? Okay. Now, opsin, where do you have this opsin and what is this opsin? Is this a neurotransmitter released by the rods? Is this a protein bound to the retinal that activates phototransduction cascade? It is a type of sodium channel in photoreceptor, or is this an enzyme that hydrolyzes CGMP? Not a difficult question, but words are interspersed and somehow put in different orders. Right. So this we know is the neurotransmitter. Neurotransmitter is glutamate. Is this a protein which is bound to the retinal? So opsin in rod is called rhodopsin. Rhodopsin's activation center is retinal. So this is what activates the phototransduction cascade. Okay. Right. Moving on. Now we did about black spot, and from here we'll start about cataracts. Right. So we know what blind spot. What is a black spot? So if you see a black spot in islands, what does it mean? Is this correct? You should see black spot, or do you think it's a problematic situation? Okay, so this is an early side of cataract formation, right? See, the term itself tells you what the issue can be. It says black spot. Spot is anything aggregation, right? Any such blob that you see. So if there is a spot on the lens, it's basically hindering the pathway of light. And that is when you have cataract, right? So if I have to define this, so in your cataract, what happens is that your uh, basically your crystalline proteins, they are very systematically arranged. Now, if the crystallines are not properly arranged, they lead to this aggregation. They are supposed to be aggregation, yes, they are supposed to be coming together, but they are supposed to come in an orderly manner. If they just aggregate in haphazard manner, it leads to their random aggregation, which sometimes uh, like they look like blob, and that gives this as a spot on the lens and it hinders the light movement. So that's why it's called the black spot, right? So in contrast, a healthy lens should have uniform, transparent crystalline proteins. So there is no disruption in the pathway of light. Okay, next question. So how do you then explain cataract? Read the options and whichever feels the closest to cataract here. Right. So as you would have already, we have discussed it so far, cataract is basically clouding of the eye lens, which is again due to protein aggregation. Which protein aggregation? Alpha crystalline. Now, which of this is the best description of cataract? Now we know it's a clouding lens. So what makes it cloudy? Is it because the lens have become shrunk, swollen, clear? Of course not, but it's still an option, clear, or it becomes blurred. Just because we call it cloudy, does it really mean that it is blurred, or it can be any other reason also? Right. This is also fairly simple. It's cloudy lens because it is lens have become blurred because of your scattering of our light. because of the spots that are now because of the aggregation of the proteins in the lens. Okay. Okay. Now, if you are to do cataract surgery, what is removed in a cataract surgery? Easy, but read and then try to answer it. Yes. So the answer will be crowded lens because cataract is a problem associated with lenses, right? So you remember we saw that this is cut open and this lens is removed, and the professionals or the doctors then introduced a artificial intraocular lens, which is clear, and that is then functioning in place of your natural lens in the eye. So your clouded lenses are removed, and what you uh, put or what you do as a correction is you put intraocular lenses, right? And that function properly. Okay, we are about to end it. Just two more questions. So the next question is, so lipoxygenase in the eye is implicated. Remember lipoxygenase we did in one of the first few questions, right? And if you can remember and recollect what did we use it from there, and then answer this question here. Look at the options and try to remember where did we read about lipoxygenase. Okay, let me give you the answer. Cataract formation by promoting protein aggregation, right? So, what is this enzyme? Otherwise, the name suggests like lipoxy. So, it's basically an oxidative, responsible for oxidative metabolism of fatty acid lipids, right? So, what does it do in your eyes? Why is it promoting or why is it facilitating cataract formation? So, if in the lens you have the activity of lipoxygenase, the activity of the enzyme can lead to oxidative stress. And this oxidative stress, as we know, they promote the aggregation of alpha crystalline. And aggregation of crystalline protein leads to cataract formation. So it basically is affecting the proteins, crystalline proteins, and then via the aggregation of crystalline proteins, it is affecting the cataract. So why are they at all going for? Yeah. And coming to the last question, sorry. Yes. So when we say eye donations, what part is generally used for eye donation? So all this so far all about eye correction and all, but we also know people uh, donate eyes. So which one do you think is used for donation of eye? Which part is generally, taken from the donor? So it's basically the cornea. So if you are talking about eye donations, cornea is basically taken from the donor and then given to the recipient, right? Well, that brings us to an end to today's problem solving session. And next week too, around the same time, we will be moving on to the lecture series of week 10. And we'll move on to the other system that was discussed in the by the professor in the animal physiology course, right? So see you in the next class. Thank you so much for looking at the video. And hopefully, if you have any doubt, please do feel free to reach out to me, post it in the comment section or the chat box, and I am there to help you. Thank you so much.